안녕하세요. 저는 CST 디스트리뷰터 매니저 김선영이고요. 오늘 저희 CST 사이언티스트가 진행하시는 웨비나에 참여해 주셔서 감사합니다. 사실 오늘 웨비나에 참여해 주신 분들이 이제 카 연구를 하시거나 이제 관심이 있으신 분들일 텐데요. 이제 카 연구를 위해서 이제 다양한 안티바디를 사용해 오셨을 거라고 생각을 합니다. 그래서 이에 따라서 저희가 2022년에 그 CST에서 범용적인 카 링커 안티바디를 타겟으로 카 안티바디를 타겟으로 이제 안티바디를 개발을 해서 처음으로 2022년에 시장에 내놨고요. 그 관련해서 지금 슬라이드에 보시는 것처럼 저희가 혁신상을 이렇게 수상을 했습니다. 그래서 올해는 또 다른 상을 수상을 했는데요. 이제 한국 과학자분들께서 이제 저희 안티바디를 사용을 하셔서 인용을 하신 후에 이제 논문을 내셨기 때문에 이제 그에 관련해서 안티바디 서플라이어 석시딩 인 사우스 코리아에서 관련 상을 받았습니다. 그래서 이 자리를 빌어서 감사하다는 말씀을 또 드리고 싶고요. 그래서 오늘 저희 CST에서 제품을 직접 개발하신 CST 사이언티스트이신 에므릭 박사님께서 이제 카 링커 제품에 대한 발표를 해주실 건데요. 이제 간략하게 이제 양력을 소개해드리면, 이제 암릭 싱 박사님은 현재 이제 저희 면역학 CST에서 면역학 제품 설계와 전략 개발을 위해서 이제 선임 연구원으로 재직 중이시고요. 이제 보스턴 대학교 의과대학에서 이제 생화학 및 분자 생물학 박사학위를 받으신 후에 이제 CST에서 14년 이상 항체 검증 연구를 진행을 해 오셨었습니다. 또 현재는 세포 치료 연구를 가능하게 하는 항체와 분석법 개발에 주력을 해서 연구를 진행을 하고 계시고요. 영어로 저희가 진행을 할 건데요. 이제 암릭식 박사님께서 질문이 있으신 경우에는 저희가 마지막에 이제 슬라이드가 다 끝난 후에 질문을 해주셨으면 감사하겠고요. 채팅창에 혹은 구두로 문의를 주시면 됩니다. 영어로 하실 분은 직접 채팅창에 쓰시거나 영어로 말씀을 하셔도 되고요. 그다음에 한국어 사용하실 분들은 그냥 말씀해 주시면 돼요. 제가 중간에서 한국어에서 영어로 통역을 해드리고 커뮤니케이션에서 도움을 드리도록 하겠습니다. 그래서 평소에 이 카 링커 안티바디에 대해서 궁금하셨던 부분, 이제 개발자가 직접 나오셨으니까 좋은 기회로 활용을 해주시면 굉장히 좋을 것 같습니다. 감사합니다.
Good morning, welcome, and thank you all for attending. My name is Amrik Singh, and I lead efforts within the product design and strategy team at Cell Signaling Technology to develop antibodies and assays to facilitate cell therapy research. It's a pleasure to be able to speak to you this morning and to represent Cell Signaling Technology to talk to you about two novel monoclonal antibodies that we have recently commercialized for the characterization of CAR-engineered immune cells. So my talk today will be divided into three major sections. The first being the rationale that guided the conceptualization and design of a new class of CAR detection reagents known as CAR linker antibodies. I'll then highlight the utility of these antibodies for monitoring CAR expression in a flow cytometry assay. And then I'll conclude the talk with a discussion focused on how these antibodies can be leveraged in additional workflows to characterize CAR-engineered immune cells. To help set the stage for this discussion, I want to first show this diagram, which I believe nicely illustrates at a very high level the workflow that is often leveraged for the ex vivo engineering of CAR T cells. To the left, it first starts with the identification and validation of antibody-based binders of a designated target antigen, most commonly a tumor antigen. And these candidate binders ultimately need to be converted into an scFv format. Moving clockwise along this diagram, transgenes which encode these candidate scFv binders are then spliced in frame into a viral expression vector, which contains a CAR backbone. And this is in order to generate viral particles for delivery of the CAR transgene into activated T cells. The resultant CAR T cells are then expanded and characterized ex vivo using a variety of assays to interrogate both the identity and potency of the engineered cells. One of the assays that is used in this panel of tests to characterize identity and potency focuses on the monitoring of CAR expression. And it's a fundamental step in the life cycle of CAR T cells, ranging from the discovery of novel CARs to clinical manufacturing, and then ending with post-infusion immune monitoring in patients. Therefore, having detection reagents that are both reliable and versatile is paramount not only to accelerating CAR research, but also to the clinical development of CAR-based cellular therapies. The field of synthetic biology has really exploded over the last few years, and it has equipped cell engineers with more tools than ever before to enable the development of novel CARs and gene circuits that are capable of rewiring T cell biology. And this is ultimately with the aim of developing therapeutic cellular platforms with enhanced persistence and effector functions. The evolution of CAR design comes about largely through the iterative splicing of many structural and functional domains that are derived from different immune receptors. And in order to interrogate the functionality of these novel CAR designs, one needs precise reagents and coupled with analytical platforms in order to address questions such as, is the CAR expressed properly on the cell surface? And is this CAR or CAR molecules competent to drive T cell effector functions, such as cytotoxicity, proliferation, and cytokine production? As you'll see in the coming slide, there are many types of affinity reagents that can be leveraged to detect CAR protein expression, each coming with their own set of advantages and disadvantages. Part of my goal today is to highlight a new class of CAR detection reagents that circumvent some of the disadvantages that exist with pre-existing reagents that are commercially available and custom made. So shown here is the prototypical structure of an scFv-based CAR. There are as you can see from the slide, there are multiple approaches one can use to detect the presence of a CAR transgene in engineered cells. Genomic and transcriptomic methods, while sensitive, lack the ability to inform one of the status of CAR protein expression and functionality. As I alluded to earlier, CAR protein expression is most often interrogated through the use of flow cytometry assays, and it's a critical attribute that is measured at multiple stages during the life cycle of CAR engineered cells. In existing reagents that enable the detection of CAR protein expression, such as the ones listed in the blue box here, are designed in such a way that they're centered on the structural elements within the extracellular domain of the CAR molecule. And this is because they're designed to be used to interrogate CAR surface expression. However, some of the drawbacks to these detection reagents are centered around the fact that not all CAR designs incorporate the structural elements that they target, or that they target structural elements that coexist on naturally occurring immune receptors. And this introduces an element of non-specificity. There's also an issue of lack of commercial availability. In certain reagents, such as anti-idiotype antibodies, while often considered the gold standard because of their high sensitivity and specificity, can only detect a CAR of defined specificity, and thus essentially need to be custom generated for each unique scFv. Other commonly used reagents, such as Protein-L, suffer from the fact that they're not easily incorporated into multi-parametric flow panels. Therefore, there's a need for a more universal CAR detection reagent, in particular, one that will enable a greater degree of efficiency for expression screening during discovery phases. And this is when optimization of CAR structure, including that of the scFv, is likely to occur. I believe this will be particularly impactful for CARs that are directed towards novel experimental antigens. Therefore, several years ago, we asked whether there are other structural elements within the CAR structure that can be exploited to create novel detection reagents. CARs that utilize an scFv for target recognition are the most widely used in research and clinical settings. And the linker sequence that are used in these scFvs is critical not only for the stability of the CAR molecule, but also for the proper alignment of the variable heavy and variable light domains, so that they can form a competent conformation for target antigen binding. And this also prevents spurious aggregation of the domains. Most scFv-based CARs utilize linkers that have multiple repeats of 4 glycine and 1 serine, or a sequence of amino acids known as a Whitlow peptide. Because these two linker sequences are fairly ubiquitous in scFv-based CARs, we sought to raise monoclonal antibodies against them in the hopes of identifying clones that can detect a broad range of scFv-based CARs which contain these linkers. And indeed, we identified clones against each unique linker sequence as illustrated here. These clones are highly versatile and, as I have alluded to, can detect a broad range of scFv-based CARs agnostic of antigen specificity, making them ideal tools to use in high-throughput flow screens for CAR expression. So in a sense, when these two clones are combined together in screening assays, they can be classified as universal detection reagents. And I'll highlight the performance of these clones in a flow cytometry assay in the next section. These clones can also be used to interrogate surface expression of CARs on multiple types of cellular as well as logic-gated platforms that are currently being leveraged in both discovery and clinical studies. Lastly, I want to point out that these clones can be plugged into multiple workflows. For example, we have generated an expansive portfolio of conjugates for each of these clones, which facilitates their incorporation into high-dimensional flow panels. Furthermore, the clones can also be used for enrichment and activation of CAR-engineered cells, two utilities that I'll describe in more detail towards the end of the talk, which allows one to interrogate on a deeper level the biology of CAR-engineered immune cells. So how do these antibodies perform in a flow cytometry assay? To validate the anti-linker clones in a live cell flow cytometry assay using both relevant and conventional model systems, what we did is we generated CD20 CAR lentiviral constructs, which contain either a Whitlow or G4S linker embedded within the Lu16-based scFv. These bicistronic constructs allow for not only for the co-expression of the CAR, but also of tagged BFP in virally transduced cells. And the analysis of BFP expression allows one to identify the retrovirally transduced population, which should also express the CAR of interest. Viral particles carrying these constructs were then used to transduce human T cells prior to analysis of CAR expression by flow cytometry. Looking at this dot plot of the CD20 CAR containing a Whitlow linker, one can clearly see that staining with the anti-Whitlow linker clone highly correlates with BFP expression in primary human T cells, and we observe little to no staining of non-transduced T cells. And to further confirm the specificity of this clone, we stained a more complex cellular matrix in the form of non-transduced human PBMCs, and we observed no spurious cross-reactivity in any of the immune cell populations that exist within this matrix. An identical validation approach was employed for the G4S linker clone, and we show in the series of dot plots on the right-hand portion of the slide that this clone is able to specifically detect a G4S linker containing CAR in primary human T cells. And similar to the Whitlow linker clone, we observed no spurious cross-reactivity in any of the immune cell populations that exist within non-transduced PBMCs. We next wanted to compare the sensitivity and specificity of the G4S linker clone to other established detection reagents for CD19 CARs, which contain an FMC63-based scFv. And in order to do this, we created a stable cell line expressing a CD19 CAR with an FMC63-based scFv and a G4S linker. And the detection reagents shown here displayed no staining of non-transduced Jurkat cells. I also want to highlight that the Whitlow linker clone does not cross-react with the CD19 CAR containing a G4S linker. More notably, however, the staining efficiency of the G4S linker clone compares favorably to that of the anti-idiotype antibody as well as the CD19 target antigen in cells expressing the CD19 CAR. We took a similar approach with the Whitlow linker clone, comparing its sensitivity and specificity to other established detection reagents for BCMA CARs, which contain a C11D5-based scFv. And for those of you who are familiar with the 6 FDA-approved CAR T therapies, the C11D5 scFv is the binder that's used in Abecma, which is one of 2 FDA-approved anti-BCMA CAR T cell therapies. Getting back to the data, the detection reagents shown here displayed no staining of non-transduced Jurkat cells. In the G4S linker clone, I also want to point out that the G4S linker clone, similar to the Whitlow linker clone, does not cross-react with the BCMA Whitlow linker CAR. And more notably, the staining efficiency of the Whitlow linker clones compares quite favorably to that of the anti-idiotype antibody and the BCMA target antigen in cells that are expressing the BCMA CAR. In addition to I want to switch gears a little bit here, but still focusing on using these antibodies for detection of CAR surface expression. In addition to redirecting the specificity of T cells with CARs, there's a strong desire in the field to express CARs in other types of immune cells, such as NK cells, monocytes, and macrophages. The FC domain of rabbit IgGs is known to bind to human FC receptors, and this property of rabbit IgGs could potentially lead to false positive staining when using the linker antibodies as a standalone staining reagent for interrogation of CAR expression on immune cell types such as NK cells and monocytes. Therefore, what we did was we generated FAB prime 2 fragments of each of these clones. And as I'll show you in the coming slides with some data, supportive data, the specificity and sensitivity of these FAB prime 2 fragments is comparable to that of the full-length whole IgG conjugates in live cell flow assays. And removal of the FC domain alleviates the need for FC block in flow protocols. And as I'll show you, we validated these fragments for lack of FC receptor binding. And then I just want to point out that these fragments are ideally suited as standalone expression screening tools in cell platforms, in CAR engineered cell platforms that are rich in FC receptors. And these cell types could include macrophages, NK cells, and monocytes. So as I alluded to earlier, we asked whether we could remove the FC domain from the whole IgG to generate FAB prime 2 fragments of the linker antibodies, therefore eliminating non-specific binding to FC receptors while still preserving the ability to engage the linker epitope within a CAR scFv. And to get at answering this question, we first co-stained human PBMCs with a CD14 antibody. And this was done to identify the FC receptor containing monocytes within the PBMC matrix. And then in addition to the CD14 antibody, we co-stained with the whole IgG format of the linker antibody or the FAB prime 2 formats. In the scatter plots shown here horizontally clearly demonstrate that the FC domain of the whole rabbit IgG format non-specifically engages with FC receptors on monocytes. And this is indicated by the population of cells within the red squares. And then we go on to show that this non-specific binding is abrogated with the FAB prime 2 format. And we also observe abrogation of this non-specific binding with the rabbit isotype control. We followed this up by demonstrating that the FAB2 prime fragments, that the FAB prime 2 fragment formats of both the G4S and the Whitlow linker clones are able to detect an scFv-based CAR containing the appropriate linker. And this is with a sensitivity that's comparable to that of the corresponding whole IgG format. We next asked whether these clones could be leveraged in other applications to help facilitate a deeper understanding of CAR T-cell biology. Having established that they performed well in a live cell flow cytometry assay, we reasoned that these clones could potentially be incorporated into an immunoaffinity enrichment protocol. It's well known in the field that ex vivo manufacturing protocols result in a mixed population of unmodified, non-specific, as well as CAR-modified redirected cells. Donor variability, variability of transgene delivery methods, and the size of the genetic payload can all combine to result in a wide range of CAR-positive cells within a given population during the manufacturing process. And this undoubtedly can confound the interpretation of data from ex vivo functional assays, as well as in vivo studies. Therefore, we sought out to develop a column-free, bead-based immunoaffinity enrichment protocol, which is illustrated in the diagram here on the left. At a very high level, I want to point out that this protocol uses magnetic beads, which are conjugated to streptavidin via a cleavable linker. And in combination with the biotinylated CAR linker antibodies, we are able to achieve positive selection of CAR cells from a mixed population. And this is especially impactful in settings where the CAR transduction efficiency is low. Furthermore, using this protocol, we've been able to derive enriched cells that are highly pure, viable, free of beads, and most importantly, are available to be used in either more extensive flow cytometric analysis or functional assays. Now I want to show you some data showing how leveraging this protocol can generate pure, highly pure and functional CAR T cells. So we first incorporated the G4S linker into the protocol that I just described, and we were able to obtain a highly pure population of CD20 CAR T cells, starting with 11% in the pre-enrichment population and enriching up to 98% post-enrichment. We were also able to achieve a highly pure population of BCMA CAR T cells using the biotinylated Whitlow linker antibody, starting with roughly 9% in the pre-enrichment population and deriving a 97% pure post-enrichment population. We next wanted to address the functionality of the purified cells by leveraging an ex vivo co-culture assay to interrogate antigen-dependent CAR T cell activation. And to do this, what we did is we mixed CD20 CAR T cells purified with the G4S linker antibody and mixed them with antigen-positive Dowdy cells or antigen-negative Jurkat cells, and then looked for upregulated expression of CD69 and PD1 activation markers on the CAR T cells in response to targeted antigen engagement. PMA and inomycin is shown. PMA and inomycin treatment was included here as a positive control for T cell activation. The scatter plots shown here clearly demonstrate that when CD20 CAR T cells are cultured with target antigen positive cells, they robustly upregulate the expression of CD69 shown along the top row and PD1 along the bottom row, compared to when the CAR T cells are grown alone, in media alone, or with target antigen negative Jurkat cells. Continuing to leverage this co-culture assay, we extended the functional analysis to interrogate effector cytokine production. Intracellular cytokine staining demonstrated that when CD20 CAR T cells are co-cultured with target antigen positive cells, they, similar to the activation marker, the surface activation markers, they robustly upregulate the expression of TNF alpha, which is shown along the top row, and interferon gamma along the bottom row, compared to when the CAR T cells are grown in media alone or in the presence of antigen-negative Jurkat cells. Switching gears to the final section of my talk, I'd like to highlight another attribute of these clones, and that is their ability to activate CARs. As the diagram on the left illustrates, and it's not important to really focus on the details, each of the discrete signaling nodes here. But what I want to highlight here is that CARs have the capacity to engage a complex network of intracellular signaling cascades to drive immune cell effector function. Adding to this complexity is the fact that different structural elements within the CAR can dictate signal strength, as well as which signaling nodes are used to engage immune cell effector function. One challenge that has been presented with the field is for the identification of optimal CAR designs. Sorry, let me back up. One challenge for the identification of optimal CAR designs has been the lack of good model systems to investigate the impact of CAR-based stimulation. In part, this challenge has been presented to the field because of when using target cell-based assays, readouts of CAR-specific signaling can be confounded by signaling which is mediated by other T-cell surface molecules that are engaged upon interaction with target cells. Therefore, in a proof of concept experiment, we wanted to address whether or not the G4S and Whitlow linker clones could be leveraged as tools to activate CAR T cells. To answer this question, we leveraged the plate-bound antibody stimulation assay, in which we coated wells with no antibody, isotype control, the anti-G4S linker antibody, and OKT3 as a positive control inducer of T cell activation. We then added non-transduced Jurkat cells or Jurkat cells stably expressing a CD20 CAR with a G4S linker to these wells in order to interrogate T cell activation using flow cytometric detection of upregulated CD69 surface expression as a readout. As shown in these scatter plots, only the plate-bound OKT3 is able to induce CD69 expression in non-transduced Jurkat cells. However, in addition to OKT3, plate-bound anti-G4S antibody is able to induce significant expression of CD69 in Jurkat cells, which are stably expressing a CD20 CAR with a G4S linker. We leveraged an identical experimental setup to interrogate the ability of the Whitlow linker clone to activate CAR T cells. Again, we show that only plate-bound OKT3 is able to induce CD69 expression in non-transduced cells. However, both plate-bound OKT3 and Whitlow linker antibody can induce a robust upregulation of CD69 in Jurkat cells, which are stably expressing a Whitlow linker containing CD20 CAR. So in summary, I hope I've conveyed to you that scFv-based CARs can contain ubiquitous peptide linkers, namely the G4S and Whitlow linker, that can both be exploited for the development of tools which can be used to manipulate and characterize CAR-engineered cells. We at Cell Signaling Technology have identified 2 novel clones against epitopes in each of these 2 linkers, and have demonstrated that they can be used as versatile tools to enable the identification of CAR-engineered immune cells independent of antigen specificity. These clones can also be leveraged as I showed you here, they can be leveraged for bead-based sorting of CAR-engineered cells. But our customers have also reported to us that they work quite well for FACS-based sorting of CAR-engineered immune cells. And I've also demonstrated that they can be used for the selective activation of CAR-engineered immune cells. We've made each of these clones commercially available to the cell therapy research community, and we offer them. We have a wide offering of direct conjugates to a wide variety of fluorophores in both whole IgG and Fab prime 2 formats. We also offer them as direct conjugates to biotin, as well as unconjugated carrier-free format. And there's also an opportunity to work with us to custom conjugate each of these clones to functionalities which we may not offer in an off-the-shelf format. So with that, I'd like to conclude my talk and thank you to the individuals on the left of the slide who were instrumental in a lot of the technical work that went into generating these antibodies and characterizing them in a variety of different assay formats. And then to my collaborator, Jason Lohmiller at the University of Pittsburgh, who collaborated with us in developing a lot of the CAR constructs that I showed in the data today. So with that, I will conclude, and I would be more than happy to answer any questions that have come up throughout the presentation.